Hi, everyone. Welcome. Today we have a presentation from Nirvik Sinha, who's a graduate student who is going to present his work from Hebrew University in Jerusalem. Nirvik is a fifth-year PhD student in the neuroscience program at Northwestern and Hebrew University. He's mentored by Yifat Prout and Jules de Wald. You know Yifat's work from the work that she's done on control of reaching movements and the neural basis of motor control in the spinal cord. Today we're going to hear from Nirvik regarding the work that he's been doing in disrupting the cerebellar input to the motor cortex and looking at its effects on control of reaching. Nirvik received his master's degree from Indian Institute of Technology in Kharagpur and Bachelor of Medicine from Calcutta University. He's recipient of a Israel Council for Higher Education PhD scholarship, and it's really wonderful to see this interaction between Northwestern and Hebrew University. Thank you so much, Nirvik, for being here.
Thank you very much, Reza, for inviting me to the presentation. So today I'll be talking about the disentangling acute motor deficits and adaptive responses evoked by the loss of cerebellar output. So this is a part of the collaborative work, as Reza introduced, between Northwestern and the Hebrew University of Jerusalem. And I'm being co-supervised by Dr. Yifat Prout and Dr. Julius de Wald. All right. Okay. Okay, so the ability to coordinate well-timed movements greatly relies on the cerebellum. The cerebellum is thought to contribute to this function by storing an internal model through which the sensory consequences of movements can be predicted and the commands required for achieving specific motor goals can be calculated. This has been a whole field of work done in this particular concept of internal models, mainly by the work of Wilbert Kawato, Timothy Ebner, Reza himself, and his colleague Musa Ivaldi. And this kind of supports the results that have been found so far from previous studies in clinical patients, that is individuals with cerebellar deficits, where they observe that normally the reaching, which is pretty straight and consistent. In case of cerebellar individuals with cerebellar deficits, the trajectories become very much curved, chaotic, and also it's associated with a lot of errors like endpoint errors, overreaching or underreaching. And this is mostly work done by Amy Bastian and her colleagues and several others as well, which have shown this consistently in cerebellar patients. So the ability to coordinate greatly requires the prediction of mechanical interactions between adjacent joints. So this is particularly important or useful in multi-joint movements where you're controlling simultaneously two joints or more at the same time. And you need to be aware not only of the movement of a single joint, but also the movements of the linked joints or associated joints. So to illustrate this or to understand this better, consider the upper limb as a two links object. That is, you have the forearm and the upper limb. So you have the forearm and the upper arm, and they are basically moving in the horizontal plane. So you have flexion extension at the shoulder and flexion extension at the elbow. So the movements are confined to the horizontal plane. So in this kind of movement, consider the movement over here, where there is an active shoulder torque that is being applied, that is shoulder flexion. So the muscle at the shoulder are contracting the shoulder flexors to move the shoulder in the direction of flexion. So what happens is as the shoulder flexes, there is an automatic passive extension of the elbow. So this is not happening because there is also an additional extension force applied on the elbow, but it's just from the communicated or passive forces that is being communicated from the movement of the elbow. So the fact that the elbow is linked to the shoulder produces a kind of passive or artifactual force at the elbow joint. So this is the coupling torque. So this is how joints interact, and the ability to coordinate between joints is a crucial factor in multi-joint movement. So while you're controlling one joint, you have to be aware that the other joint is moving and is being affected by the movement of the active joint. So cerebellar deficits, so this has been classically shown in cerebellum that there is a main problem in coordinating between joints. And this kind of joints or coupling torques that are generated because of the movement of one joint that affects the other joint has been thought to be a principal deficit. However, cerebellar deficits extend actually much beyond motor timing. So there are several studies that have delineated sensory deficits in the cerebellar patients, like proprioceptive deficits. And another problem that has been observed in very early studies but has been ignored subsequently is the presence of hypotonia and weakness. So there's been early in the early 20th century, there was a number of studies by Gordon Holmes where he showed, like, in acute cerebellar patients, that is in individuals with acute injury to the cerebellum, mostly due to gunshot injuries, had this phenomenon of weakness in the upper limbs. So when they are, for example, here what is showing is that when they try to, they are asked to extend their upper limb in the horizontal direction, they are unable to do so. So they are affected by the gravity on the affected side, that is the ipsilateral side. And likewise, there has been also studies in non-human primates where, as well as other primates, where they have shown that, like, when the cerebellar output is disconnected, that is, there is acute sectioning of the cerebellar peduncles or a hemicerebellectomy. Likewise, the non-human primates or the monkeys also show similar weaknesses. But this finding was subsequent studies, it got buried in favor of the much more timing and coordination role, which is a lot of the studies in the later 20th century has been focused on that. So one of the issues with clinical studies is that they are unable to separate between primary and secondary deficits or effects. So what I mean by that is essentially what are the core abnormalities and what are arising as a result of adaptive responses to the primary deficits. Because mostly the clinical patients are in a chronic stage, so it's difficult to delineate what has been the incipient response as a loss of the cerebellar loss of cerebellar signals compared to what has been an adaptive responses. And also added to that, the problem is there is a lot of heterogeneity in terms of the lesions, locations of the lesions. Most cases, the cerebellum itself is not only affected, but there's also associated lesions in the cortex. So that kind of also confounds or complicates the interpretation of the results. So given this background, we asked, what is the sequence of events that produce motor impairments following the loss of signals? And to study that, we have this approach in our lab where we look at horizontal planar reaching in the keen arm exoskeleton. So for this specific study, we trained 4 monkeys to perform planar reaching tasks wearing or being supported by the exoskeleton in the horizontal plane. So the schematic of the task looks something like this, where the monkey is seated in the keen arm chair and is performing center out reaching movements to peripheral targets from the center. And the order of the task is something like this. So there is initially a central cue to which the monkey locates the hand, after which a peripheral cue which identifies which target the monkey is supposed to move to, and following which there is a delay period. The monkey has to hold the movement and is not allowed to go yet. And then the central target disappears, indicating the monkey a go signal, and the monkey is allowed to reach and then gets a reward. So the paradigm for studying this in here, like, we have certain directions where the movements are dual joint, that is involve both the shoulder and the elbow, like these targets 2, 3, and 4, and 6, 7, and 8. And at the same time, we have also single joint movements, or primarily single joint movements, where mainly the shoulder is moving while the elbow is stationary. And this can be understood better by looking at the net torques in different directions. So net torques is the torques that is resultant or proportional to the joint accelerations. So if the joint acceleration is 0 or the joint is not moving, the net torque is 0. So on the x-axis, what you have is basically the normalized shoulder torque that is normalized to the maximum torque across all directions. And y-axis is the normalized peak elbow torque. So here you can see that these targets 6, 7, and 8, and 2, 3, and 4, involve extrusions or movements, either flexion. So positive is flexion and negative is extension. It involves flexion and extension in both the joints. So it's along the diagonal over here, so there is either shoulder flexion with elbow extension during the forward reaching, or there is shoulder extension with elbow flexion during the retrieval or backward reaching movements. Whereas for targets 1 and 5, this is in the horizontal plane. The lateral targets, you mainly have movements in the shoulder. So the elbow is stationary there. So in this way, we have certain targets where there is multi-joint or dual joint movements compared to certain targets where it's much more simpler, or you have to only control one joint, single joint movement. So while the monkey performed this task, the manipulation, the experimental manipulation that we do in our lab is we block the cerebellar output by a chronically implanted electrode at the superior cerebellar peduncle using high frequency stimulation. So it's a kind of a reversible electrophysiological block by which we can study the movements during control and the cerebellar block within the same monkey. So to give you a rough idea or the background of the way that we implement the block. So the pathway that we are targeting is basically the cerebello-thalamocortical pathway. So the cerebellar output crosses the midline and innervates the contralateral ventrolateral thalamus, which is the motor thalamus or the cerebellar receiving thalamus, from which the output goes to the motor cortex. And we are essentially targeting just the output or the white matter tract from the cerebellum, the superior cerebellar peduncle. And previous studies in our lab have shown that this pathway is an efficient recruiter of the motor cortex. And when we have low frequency stimulation or stimulation at very low frequencies at 3 Hertz, we get stimulus locked responses, consistent responses. So over here, what you're seeing is a raster of the multi-unit responses aligned to the stimulus at the cerebellar peduncle. And you get consistent from stimulus to stimulate, you get consistent responses. Whereas when we switch to the high frequency stimulation or 130 Hertz stimulation, these stimulus lock responses are lost. And here is basically a PSTH showing the same thing averaged across successive or multiple stimuli. And relative to the single pulse stimulation, which elicits a very nice stimulus lock response, we have the complete extinction of these responses when we switch to high frequency stimulation, which shows in a way that we are efficiently able to block this pathway in a reversible electrophysiological manner. So while the monkeys perform these movements, we began by examining the kinematics during control and cerebellar block trials. So we found that movements exhibited a significant reduction in the peak hand velocity, as would be expected from cerebellar block. So here is an example where we are seeing a profile of a single trial peak velocity of the hand, that is the endpoint tangential velocity during control in blue and cerebellar block in vermilion. So what we wanted to see is, given that there are certain directions where the movements are multi-joint and involve reaching compared to certain directions that the movements are single joint, or there are movements in the backward direction, which is a retrieval movement, we wanted to see how the hand velocity was affected across different targets. So we found that the effect of the cerebellar block was target dependent on the hand velocity. So here what you're seeing is basically a polar plot of the change in hand velocity following the cerebellar block. So each point is basically a percentage change relative to the control. So we have certain directions, mostly the reaching directions, targets 1 to 4, which have high reduction in the velocity. So the radial direction is the percentage reduction. Whereas for certain directions, the retrieval directions, the reduction is not apparent or sometimes also not significant. So the question that we asked, is that why is there this asymmetry? Like normally we would expect that all targets to be affected or not, but we found this asymmetric response in different targets. So in order to try to understand this phenomenon, what we did is the question that we asked, that is, how can we explain the target dependent effect of cerebellar block on the hand velocities? So what we did is we performed an inverse dynamics model of the upper limb to identify the target specific muscle and coupling torques. So by coupling torques, I mean the interjoint interactions, so the torques that are passively generated as a result of active muscle forces on the attached joint or linked joint. So, to understand this model on a brief basis, consider again the upper limb as a 2 links model moving in the horizontal plane. So the forces acting at each individual joint can be represented by this equation, which is presented in a simplified manner. So the terms here are basically the muscle torques, that is the torques that are produced as a result of active muscle forces and passive viscoelastic interactions at each individual joint. And then there is the net torque, which is basically the resultant or what you see at the joint that is proportional to the joint acceleration. And then there is the interaction torque or the coupling torque that is arising as a result of interjoint interaction. So these are torques that are velocity and acceleration dependent, as well as dependent on the inertial properties of the limb, that is the attached limb. So the faster you move, the greater are the stronger are these interactions. So these basically torques are velocity and acceleration dependent. So the muscle torque is essentially a resultant or a difference between the net torque and the coupling torques. So using this model, we are able to obtain or tease apart the essential components of torques that are being applied at each joint and resulting in the hand movements. So we can identify the muscle torques and the coupling torques. So we, to illustrate to you an example, so consider a movement in the forward direction, that is target number 3. So here on the left-hand side is a plot of the shoulder torque of one trial. So there is an active movement of the shoulder flexion. So positive is flexion. So the monkey generates an active shoulder torque. And as a result of this movement, as I said earlier, there is a passive extension torque. Because the shoulder is flexing, the elbow is extending passively because of the action of the shoulder. So, and what you see is because the elbow is moving, there's again a feedback at the shoulder due to which there is a passive flexion torque also at the shoulder. And it happens so that in these movements that the passive torque at the elbow is sufficient to drive the elbow joint. So the muscle torque is almost 0. So here you can see another, again, an example of how you need to coordinate between joints. So when you're moving to target 3, normally you would expect to flex your shoulder and extend your elbow. But because of the shoulder flexion and the larger inertial properties of the proximal limb, the elbow is automatically extending. So you don't need to really drive your elbow. So this is where this concept of coordination between joints comes. So having understood the muscle and coupling torques, now we wanted to look at the different amounts of magnitude of muscle torques that were generated across different target directions and see whether that can explain the target dependent effects on the hand velocity. So the target specific reduction in hand velocity was essentially produced by a change in the muscle torques, muscle commands. So here what you're seeing is again a similar plot that I showed you at the beginning. So these on the x-axis is basically the shoulder muscle torques normalized to the peak torque across all targets. So there are certain targets, for example, target 2 that generate high torques compared to target 4 or 8, where the shoulder torques are lower. And likewise, on the y-axis, you have the elbow torques that is normalized to the maximum torque across all 8 targets. So here again, as you can see, there are directions that have high shoulder torques or high elbow torques. And overall, what you see before considering the target specific effects, the cerebellar block that we implement in our lab, it results in reduction of the muscle torque. So the overall plot is contracted, as you can see. But if you notice specifically in specific targets, you find that the reduction in the reaching movements, that is the targets 1 to 4, where we saw the maximum reduction in hand velocities, is also where the muscle torques are maximally reduced, especially in the shoulder direction. And compare that to the retrieval movements, there is a very minimal change. That is the targets 8 through 5. So another thing to notice is that if you look, targets 2, 3, and 4 are the dual joint movements. Compare that to the target 1, which is a single joint movement. And we find that there is reduction of muscle torques also in the single joint movements, which are much more simpler. So we find that the cerebellar block produced multi-joint reduction of torques in multi-joint movements as well as single joint movements. Now, a question was that there is also the coupling torques acting at different directions. And previous studies have shown that the coupling torques can, the cerebellar individuals or individuals with cerebellar deficits often exhibit abnormalities in controlling or managing these coupling torques. So before going in that, just to show you how the muscle torques at the shoulder and elbow were affected, or like to simplify this plot over here. Here you have basically the muscle torque changes similar to the velocity plot changes that I showed you earlier. And you can again see that these the directions, the reaching directions are maximally affected. But important to notice that at the shoulder joint, there is effect also in the single joint movements, as we saw in the hand velocity plots. And comparatively, the elbow is less affected, and there's minimal changes in the elbow torque as a result of the cerebellar block. So essentially, the changes in hand velocity are mostly due to target-specific reduction in the shoulder torques, which is maximal in the reaching directions, and also equally present in both the dual as well as the single joint reaching. So now focusing on the interjoint interactions, so the coupling torques. So as we would expect, the coupling torques also varied in a target-dependent manner. So here what you're seeing is basically, again, a similar plot as the previous one, but in this case, the coupling torques or the interjoint interactions. And what you notice is that depending upon the target, the coupling torque may be high, as in the case of targets 2, 3, and 4, or 6, 7, and 8, but low, as in case of targets 1 and 5. As I said, these are single joint movements or predominantly single joint movements. So the extent or the magnitude of interjoint interactions are low. So given that the cerebellar patients from previous studies that have shown that they have an abnormality or problem in predicting these coupling torques. So the amount of coupling torque that probably would be generated in one direction or the other at the shoulder and the elbow, given that the cerebellar patients already have an abnormality with this. So we asked the question that whether the reduction in hand velocities, especially for the reaching directions, are somehow associated with the different magnitudes of coupling torque in different directions or the spatial tuning of coupling torques. So in order to understand or to associate that, we performed a correlation analysis between the extent of reduction in hand velocity to the corresponding amount of magnitude of coupling torque at both the joints in control condition. And what you're seeing here is basically the results of the correlation analysis. So on the x-axis is basically the coupling torque in control. It has been normalized to the max across all directions. And y-axis is the change in hand velocity as a result of the cerebellar block. And these individual points are basically the averaged data points generated per session across all the 4 monkeys. And they are color coded by the target direction. So the lowest changes in velocity are in the single joint case, that is target 1, although there is a significant change there. And as you move to targets with higher amounts of coupling torques, we see higher reductions in the hand velocity. So this kind of hints to the understanding that the monkeys are not being able to manage the interjoint interactions, and thereby slowing the movements to the extent that the directions that have higher coupling torques are the ones that manifest the greatest slowing of movements. And this kind of makes sense or is logical because, as I said earlier, the interjoint interactions are velocity dependent. So the more faster and ballistic your movements are, the greater the interjoint interactions will be. So if you're not able to manage or handle them, so the logical tendency is to reduce the velocity of the movement so that these interjoint interactions become less important. So having observed this, now the question, as I asked, as I pointed out in the beginning of my presentation, is how to disentangle these effects. Like we have at one hand, there is a global reduction in muscle torques, both in single as well as dual joint movements. At the same time, the hand velocity reductions are also to a certain extent correlated with the degree of magnitude of interjoint interactions. So to understand that, so basically to summarize that, so we have cerebellar block reduced hand velocities in a target dependent manner that is correlated with the spatial tuning of interjoint coupling torques. At the same time, it's also produced by systematic target and joint specific loading of muscle torques, with the point to keep in mind that muscle torques and hand velocity was also reduced in single joint movements. Now,
maybe we can ask some questions since it sounds like you're summarizing here.
Yeah.
If I understood it correctly, there were 4 targets that the monkey used to reach outward, and then 4 targets that they just simply brought the hand back.
Yeah.
And the differences that you're showing, have you tried movements that were to 8 targets rather than 4 targets, so you could dissociate return movements from simply outward movements?
So we have all 8 targets, movements to all 8 targets in this study. So you're asking if
I misunderstood. I thought you mentioned that the
Mhm.
bottom four were
Mhm.
return movements, which I assumed that was from the out back to center.
Okay, so let me go back to the figure of the movements. Okay, so here, so for all of the movements are performed basically from the center. So from the center outward or center backward or inward. So everything is starting from the center of the.
Thank you.
Yeah. Okay, so
Did
fact,
you notice any changes in reaction time other than just slowing of the movement?
Yes, so that is something that I have a little bit, I talk about a little bit towards the end of my presentation. But yes, there are effects in the reaction time, which is like we observe that there is prolongation of the reaction time in these movements. So the movement onset is delayed. So which kind of hints at the fact that the motor preparation is affected. And I will be talking towards the end of my presentation about that a little bit.
Thank you.
Yep.
Are there any other questions for Nirvik?
Yep.
Yeah, Dan has raised his hand. Go ahead, Dan.
Hi, can you hear me?
Yeah, sure.
Sorry, my camera doesn't seem to be working. Just a quick question. I guess I had understood the coupling the elbow versus shoulder coupling torques plot to make a prediction that 6, 7, 8 and 2, 3, 4 would have sort of equivalent, since they both require torque dependent coupling, or sorry, coupling, that they would both exhibit, if the effect is on coordination, then you would expect to see symmetric effects. But you're seeing very asymmetric effects.
Yes, so this is something that we have also been debating about. I appreciate that you brought up this point. So the fact that like the majority of the effect, or the greatest magnitude of effects, both in single and dual joint movements, and the correlation with coupling torques, as you pointed, is confined to these reaching directions, as contrast to the retrieval or backward movements, where also sometimes the coupling torques can be high, but we do not observe it. So right now, like we do not have a very good answer to this. Like there are some aspects which we have thought about. Like one is that like the effect of the cerebellar output has not only effects on the cortex, but also direct interactions or effects on the spinal cord. So we believe at this point, there is a couple of reasons why this might be the case. One is that there is possibility of some variability between monkeys. Like when we are targeting the cerebellar output, it's possible that we are not equally targeting all all of the fibers. The other possible explanation is that the cerebellar descending input to the spinal cord via the brainstem or directly to the spinal cord has a bias towards the flexors, predominantly the shoulder flexors, because that's where we observe the maximum deficits. But yeah, I guess further studies or like exploration is required to kind of try to tease apart why this is the case. I'm happy to if any of you guys have any feedback on this. Like so this is something that we've also been debating about, like why this is the case.
Thanks. Yeah, I just want to make sure I was understanding it correctly. The one thought that I had is maybe there'd be some way to add endpoint load to sort of increase the coupling torques. If you had a weight in the hand or something like that, might be an interesting way to increase the coordination dependent coupling. But
you're asking the right questions. Thank you. So yeah, so this again towards the end, depending upon the availability in time, like I will be speaking about the free reaching movements that so far all of our studies have been based on the horizontal planar supported movements where the weight of the limb is supported by the exoskeleton. So the monkey doesn't have to bear the weight of the limb. And right now we are running, we have done some experiments, and also we are doing additional ones where the monkey performs like free reaching movements where the monkey has to support the weight of the limb and perform the movements. And there actually we attach some loads. And as you have already pointed out, like the effects of adding the load increases, but there we feel it's not just the coupling, but also the fact that the inability to drive the joint itself, like the shoulder joint, which we feel right now is more of like the lack of power or weakness rather than the interjoint interactions. But again, that's something that I'll be talking about in the end, and we can have a discussion on that. All right. Any other questions? Okay, so I'll move on. Anyway, so in order to try to delineate these two effects, like we had an approach over here where essentially what we are trying to do is, okay, so the advantage of reversibly blocking the cerebellar output is the ability to track the temporal profile of motor impairments. So essentially, to this end, what we did is we assessed the changes in hand kinematics, namely the hand velocity, produced by the cerebellar block across successive trials. So we have blocks of trials in control and cerebellar block, right? And the monkey is performing movements to different targets. And we have the sequence in which the targets are presented, which is essentially random. So what we did is essentially preserve the order in which the targets were presented and identify target by target how the hand velocities evolve across time. So we observed or we examined the evolution of peak hand velocities in the temporal sequence of target presentation for single joint movements and contrasted that with the dual joint movements where both the shoulder and the elbow is moving, targets 2 and 4. And again, as we confined our analysis in this case to the directions where we observed reductions, that is mainly the reaching directions. And this enabled us, the reason why we did this is because this enabled us to dissociate between primary and adaptive effects. So by primary, I essentially mean is the effects that are acute in onset or onset immediately in the first few trials, and contrast them to adaptive responses that take some time or basically gradual and progressive in onset. So in order to like how we did this, in order to explain you or try to show that, like consider these blocks of trials over here. So we have like several blocks, block 1, 2, and 3, and the targets are presented in a random, uniform random order. And there are, of course, some trials where the monkey fails to perform the movements. And say we are focusing on movements to target 1. So we identify these targets and the corresponding location or order in which they were presented across all the blocks. So we get in the end essentially a matrix where we have the repetition or the number of block on one direction, and the other direction is the trial number. And we can isolate target by target movements in successive trials. So after doing this, we wanted to examine the single joint movements. So over here, what you're seeing is movements to target number 1, that is the horizontal lateral direction, whether there's shoulder flexion and the elbow is stationary. And what you're seeing here is basically the evolution of the peak velocity across successive trials performed across these blocks. This is basically averaged and the confidence interval across the blocks. And as a percentage of the peak velocity in the first 2 trials in the control condition. So the blue trace over here is the control velocity, and the red trace over here is the cerebellar block condition. So what we observed is that right from the beginning or in the first few trials itself, there is a reduction in the velocity. So it's acute in onset. And as we progress over successive trials or the successive order of the trials, the effect pretty much remains the same. So there is no change in the magnitude. So it's pretty much a consistent DC shift or reduction in the hand velocity. So this is the single joint case. And now what we wanted to do is contrast this effect or this phenomenon with the dual joint. So overall, in change in hand velocity due to the cerebellar block was unaffected by the trial sequence, or the trial order or the order in which the targets was presented. Now contrast this to the case of the dual joint movements, where you see that, so here we have pulled together the movements to targets 2, 3, and 4 because they showed similar results. So we see that initially there is a acute reduction in the hand velocity. And as we progress further across successive trials, there is a further decrease in the hand velocity. So it's a 2-stage effect in an early or initial reduction and then a successive or later reduction. So in order to just confirm this, like we group these trials into early and late trials. That is late trials where the velocity reached an asymptote in the cerebellar block case. And we confirmed this that compared to the early trials, the cerebellar block, the late trials much more lower in velocity, and the effect relative to the control also increased in size. So overall change in hand velocity to cerebellar block for the multi-joint or dual joint movements were affected by the trial sequence, with an early reduction in the acute or first few trials and then a subsequent progressive decline until it reached an asymptote. So, so in a way, essentially, this taken together, these results reveal that hand velocity of multi-joint movements during cerebellar block was already low initially, reflecting a primary deficit. And over and above this, there was a further progressive decline in the hand velocity during successive movements. And it's most likely due to an adaptive response to the inability to compensate for coupling torque. So as the monkey realizes that these interjoint interactions are difficult to handle, so there is a progressive adaptive further decline in the hand velocity because these are dual joint movements, which we do not observe in the single joint case where presumably the interjoint interactions are already low initially. So whatever is happening is right at the beginning of the trial. So now important part of cerebellar patients in the kind of motor deficit that they exhibit is essentially that has been observed in a number of the previous studies is timing errors, like decomposition of movements and increased spatial variability of trajectories. So by decomposition, I mean like when you're moving, performing a dual joint or a multi-joint movement, you need to synchronize the movements of both the joints. That is, there is a specific temporal association between the joints, and most of the cases, the joints are moving simultaneously. But a lot of work by Amy Bastian has shown that in cerebellar patients, there is a tendency to break down these dual joint movements into individual joints in an asynchronous manner or chaotic manner, which results in like asynchronous activation of individual joints that are not having any specific temporal relation between each other. And the spatial variability in trajectories is kind of a motor noise, whereby movements performed to successive movements performed have very little similarity between each other. So one movement has one kind of trajectory, the other movement has a totally different kind of trajectory. So from trial to trial, the variability of trajectories is high. So what we wanted to do is, because these measures are velocity dependent, as you might be expected, like for example, in the case of spatial variability, if you're performing slower movements, presumably you will be more consistent because of the principle of speed accuracy trade-off. So if you're slower, you're more accurate. And likewise, in the work of Amy Bastian, she has shown that slower movements also tend to be more decomposed because the way the decomposition is computed is based on absolute joint velocities. So if the joint velocities are lower, they are around the threshold for which you might define as a stationary joint. So what happens is that there it's very noisy around that low velocity, so the movements tend to be decomposed. So the question that we ask is that the combination of primary reduction in muscle torques, or what we term as weakness, and compensation for coupling torque, that is an adaptive response, the main driver of this abnormal motor timing. That is because these measures of motor timing are very velocity dependent, whether the changes in velocity can explain these timing errors that we see in the cerebellar patients. So in order to try to answer this, we examine next the measures, these measures, that is decomposition and trajectory variability during the reaching movements where the impact of hand velocity was maximum, and how they were affected by the changes in hand velocity. So coming to the movement decomposition first. So okay, so this is basically an example of a few trials, or around 80 trials for one monkey, showing the level of decomposition of movement in control condition. So on the x-axis, you have normalized time. So we took a set of trials, a bunch of trials, and normalized the time so that they were comparable between each other. And what you're seeing over here is each row is essentially a raster of either 0 or 1. So 1 indicates that the movement was decomposed. So in a way, the way we are defining decomposition is basically whether either the shoulder or the elbow joint is stationary, but not both. So if both joints are moving and both joints are stationary, the movement is not decomposed. But if either of the joint is moving, then the movement is decomposed. So in the early part of the movement, around 20 to 30% of the movement, during which the joint velocity is crossing the threshold that was fixed based on previous literature, you see a lot of decomposition, which is kind of expected because the measure is noisy over here. But for the rest of the movement, it's mostly the joints are synchronized. Either they are moving in tandem, in sync, or they are stationary in sync, but they're never like either of the joints are activated. When we contrasted it with cerebellar block condition, we observed, as would be expected from the previous studies, is that the decomposition is not only there in the early part of the movement, but also throughout the movement. So throughout the movement, either of the shoulder or the elbow joint is moving independently or isolated manner where the other joint is stationary. So what we wanted to do is that see how these measures varied with velocity. So as I said, the movement decomposition is a velocity dependent measure. If you have slower movements, the movement tend to appear to be more decomposed because the way it's being measured. So what we did is basically compare the degree of decomposition change relative to all the control trials in the cerebellar block condition and the velocity matched control trials that were matched in velocity to the cerebellar block condition. So what you're seeing over here is basically the percentage change in the degree of decomposition or the proportion of decomposition in the cerebellar block condition, which is overall increase around by 50%, which is like a very large increase in decomposition. But also the cerebellar block condition, also the control condition where the velocities are matched to the cerebellum. So these are the subset of control trials where the velocities are similar to the cerebellar block condition. So in a way, they are slower moving trials. And as we would be expecting, there is a slight increase, a significant increase, but slight, around 10% increase in decomposition. But it's no way as much as the amount of decomposition we observe in the cerebellar block. So this indicates that even at matched velocities, the movements made during cerebellar block were more decomposed than the control. So next, we looked at similar effects on the trajectory variability. So what we are seeing over here is basically the hand trajectories that have been aligned to the vertical direction. So a few examples in the control condition in the blue and the cerebellar block condition in the vermilion or red. So what we are seeing over here is basically trajectories of the endpoint made to a single target as an example. So the trajectory, in order to measure the variability in trajectory, we did it in stepwise in this manner. So first we measure the trajectory deviation as the maximum distance from the straight path, that is the straight, absolute straight direction. So on the red arrows over here shows the trajectory deviation of the cerebellar block trials. And then we quantified the variability in the trajectories as the variance of these trajectory deviations. So if the trajectory deviations are consistent in one direction, or they are absent altogether, the variance in this trajectory deviation will be less. Whereas if the trajectory deviations are widely varying, so for example, one is in clockwise directions in the other trial in the counterclockwise direction in larger amplitude, then the variance will be high. So when we measured the control trials or control trajectories with velocity matched to the cerebellar block condition, as would be expected by the speed accuracy trade-off, the variability was 5% lower than all the control trials. So the slower movements are less variable. But in the case of cerebellar block conditions, the variability was increased despite the fact that the movements were being slower. So the cerebellar block showed 35% more trajectory variability compared to the velocity matched control trial. So in other words, despite the movements being slower, cerebellar block increased trajectory variability. So just to summarize the findings and have some concluding points over here. So our results indicate the cerebellar block leads to slower movements via two interrelated mechanisms. One is a generalized reduction in muscle torques. So when I again say generalized, it's mostly to the reaching directions, suggesting most likely what could be motor weakness because it's acute in onset and it persists throughout the movements, successive movements. And then there is a spatially tuned decrease in velocities in targets where interjoint interactions are large, that is where the coupling torques are large. And since coupling torques in multi-joint movements are generated in velocity-dependent manner, as I explained earlier, slowing down movements not only simplifies the kinetics by reducing the impact of coupling torques, but also allows more time for the slower feedback mechanisms, for example, visual feedback to aid in guiding the movement. So it no longer is essentially a feed-forward internal model-based command where you have predictions of the interjoint interactions, but you can guide your movements based on the visual feedback because you're moving slower. So our results also revealed the time course of these impairments based on examinations of successive velocity profiles of successive movements. There is an acute onset reduction in the hand velocity evident in the first few trials, following which there is a gradually emergent adaptive slowing in the subsequent trials, which is likely the adaptive response to the inability to handle or compensate for interjoint interactions. So we also observed increased motor noise in terms of movement decompositions or asynchronous activation of individual joints, which are no longer coordinated with each other, and increased intertrial trajectory variability. And our results showed that the movements in cerebellar block are more decomposed even at matched velocities, where they are supposed to be less decomposed normally. And our results also show that the movements in cerebellar block exhibit increased trial-to-trial spatial noise despite the slower velocities, counterintuitive to the speed accuracy trade-off principle. So this kind of hints at movement planning errors, because a number of theoretical studies or conceptual studies earlier have debated that the cerebellum is known to participate in planning of movements, in addition to the online control of movements, that is interjoint interactions and motor coordination and timing, by aiding the estimation of the state of the limb by integrating sensory information from its last known state with the predictions of its response to the later motor command that is being generated. So in the absence of cerebellar signals, there are planning errors in the feedforward command that orients the limb accurately towards the target. And these feedforward errors may in turn translate into noisy joint activation patterns that we are seeing here in terms of increased decomposition, and unpredictable hand trajectories, that is increased spatial or noise in the trajectories that we observe in our results. And more importantly, unlike the case of interjoint interactions, these deficits cannot be compensated for strategically. So irrespective of the hand velocity or any of the compensatory strategies, we see that this kind of internal noise that we are seeing persists throughout all range of movements in the cerebellar block condition. So overall, taken together, our results can delineate the processes underlying cerebellar dysfunction that culminate in a range of motor impairments, namely acute deficits in the ability to generate sufficient muscle forces or muscle torques, and also an inability to compensate for interjoint interactions that give rise to further slowing of movements. And then there is this phenomenon of increased motor noise, which are likely due to increased or abnormal planning of the movements. So as a segue to this, like I would end with a few things. Like one is looking at the neural activity in the planning phase, because since we see these planning errors in the movement, as also I pointed out earlier, there's increased reaction time during the movements. We want to see how the neural activity is affected in the motor planning phase. So one way of looking at it is to look at the the motor cortical oscillations during different phases of the movement. So modulations in 12 to 40 hertz oscillations in the local field potentials reflect sensory motor processing during planning and execution of movements. So this has been a number of studies that have shown that the beta oscillations, that is oscillations in these medium frequency bands, are modulated during movement, just at the beginning of the movement or even before. During the preparatory phase, there is suppression of beta oscillations, which is associated with the increased cortical excitability and planning for the upcoming movement, and then there is a rebound or increase in the beta oscillations. So the activity in the thalamocortical loop is an important source of this modulation. A number of studies have shown that. And we also know that the cerebellum provides strong inputs to the motor thalamus, as in the cerebellothalamocortical pathway. So given this background, we asked the question, and also the findings in the behavior in planning errors, we asked the question that what is the role of the cerebellar output in modulating these cortical oscillations, specifically the beta oscillations in the motor cortex during the preparatory phase of the movements. So in order to try to answer that, we examine this particular phase of the movement, that is the delay period, where the monkey is preparing for the upcoming movement but hasn't started the movement itself. So this kind of, again, is hinted from the motor planning errors. To look at it in another way is to look at the reaction time. And as I said earlier, that we observed that there is an increase in the reaction time during these kind of movements following the cerebellar block, which hints that there is something going on in the delay phase or the preparatory period that is already happening before the movement itself is happening. So in order to examine this, we examined local field potentials during control and cerebellar block trials from around 250 sites, shoulder elbow related primary motor cortex sites pooled across all the 4 monkeys. So just to keep it simple, I'll be showing the results pooled across monkeys, but they're more or less consistent across monkeys. And we examined the visual cue aligned responses in 4 to 50 hertz of LFP. So we wanted to focus on the early preparatory period from the cue to the go, where the movement hasn't begun, but the monkey knows where to move, so it's planning for the movement. And what you're seeing over here is the average local field potential response are aligned to the cue. So on the x-axis is the time in milliseconds, and y-axis is the frequency in 4 to 50 hertz. And the green line over here shows the presentation of the visual cue. So all the LFP responses are aligned to the spectral responses aligned to the cue. And since we wanted to focus on the preparatory period, I have demarcated in the shaded region over here the movement period. So this is the time point from which all the movements begin. And over here, what you're seeing is basically increased activity in the beta band in anticipation of the upcoming cue, and then subsequent decrease. So this is basically unnormalized log transform power. And in the cerebellar block condition, we found that there is suppression of this change from the increased beta activity to the reduction. So in order to quantify this, we looked at these two distinct bands of beta activity, that is the lower beta from 12 to 25 hertz and 25 to 40 hertz, the higher beta. So spectral, in order to compare between the two conditions, spectral activity was normalized to the response before the cue, roughly 200 milliseconds before the cues, because before that, the monkey doesn't know where to move. So it's kind of like a resting period for us. So over here, what you're seeing is basically the responses relative to the pre-cue activity in the beta band during the control condition and the cerebellar block condition for the high and the low beta bands, that is around 12 to 25 and 20 to 40 hertz. And what you see in both cases, especially in the low beta band, that already during the preparatory period, there starts to be a difference between the control and the cerebellar block condition. So roughly after around 200 milliseconds, which is the time for visual processing of the cue information, the trajectories separate. And in the case of the cerebellar block, the degree of suppression of the beta band is much lower compared to the control condition. And just to quantify it, we looked at the time point, which is we identify as the end of the preparatory period, which is 100 milliseconds before the kinematic, the hand velocity based movement onset, because that's when the time when the EMGs start to be active. So that's roughly, if we identify at that point, we compared the degree of beta suppression during control versus cerebellar block, and we found, as would be expected, that the degree of suppression is reduced. So okay, so now to end, I would like to show some of the new experiments that we are performing in the context of the question that Dan earlier asked. Like, so overall, in the KINAM system that I have shown you so far, the weight of the upper limb is supported by the exoskeleton. So the monkey is performing these movements in the horizontal plane, and the exoskeleton is supporting the movements. So from this, we are transitioning to now free reaching movements, where the monkey not only have to perform these planned movements to different targets, but also to support the weight of the limb as these movements to different directions are performed. So here what we have is a similar paradigm where the monkey performs center out reaching movements, but in this case, has to support the weight of the limb. And a certain portion of the movements, the peripheral target also jumps to another location, but that is a story for another day. So I wanted to end by showing you some examples where we perform limb loading or loading of the distal limb, that is the forearm, during control conditions. So I'll just switch to the other mode. Okay. So over here, what you're seeing is center out reaching movements with the weight attached to the upper limb. I don't know if it's visible. This is like the white part over here. And the monkey performing these center out reaching movements during the control condition. And then we have the movements during the cerebellar block, where we see that the main problem or the deficit is already before the starting of the movements, the monkey is unable to maintain the weight of the limb at the central target itself. Like, there's a lot of difficulty. I'll play this video again. In the ability to maintain the unsupported limb with the weight attached in the limb in performing these movements. So that's something that we have been observing. And we'll at this point think that there is this problem of not being able to perform the movement itself is related to the proximal weakness or driving the proximal muscles to the motor and premotor cortices. So to end with my presentation, I have few examples of neural activity in the primary motor and premotor cortex, which kind of supports the idea that I am projecting now. So over here, what you're seeing is the peristimulus time histogram locked to the movement onset in primary motor and premotor cortices during movements to the higher targets. Over here, you see the tuning of this neuron, which is tuned to the movements where the monkey has to lift the upper limb to reach the higher targets. And compared that to the lower targets where the monkey has to basically release the weight of the limb. So these are gravity assisted versus the gravity counteractive movements. And in both cases, we see that during preparatory period itself in the premotor cortex and the execution period in primary motor cortex, there is a suppression in activity, or this timed response in the cortex, motor cortex is reduced, which kind of likely hints towards the inability to generate sufficient muscle force in performing the movements. So that's all I have. I would like to thank my lab members, Hen and Ora, and also my co-supervisors at Northwestern and the Hebrew University for guiding me through this project. So if you have questions for discussion, I'm sorry I went a bit long with the time. So I hope we have at least 5 minutes.
Wonderful. Nirvik, that was really wonderful. And I thought the videos at the end were really illustrative. Let's have some questions for Nirvik. I guess I'll go first. The results that you showed at the end regarding holding the hand, as you very well showed, the subject has a really hard time with the added weight, in particular, maintaining the hand at the center and the weakness that arises. The cerebellum has certainly in the oculomotor literature has been shown to have a lot to say about holding still and that support that you need to hold it. And do you think this is a component of that reaction time that you showed earlier?
So you mean in terms of motor planning?
Well, if you can't hold at a particular location, it affects the reaction time. And the decision that the animal makes in terms of what movement to make. In mice, many reports show that this activity during reaction time is a component of cerebellum and motor cortical activity. Like before the animal licks, for example, the ramping activity is disrupted if you affect this, output of the cerebellum. Maybe it's something similar.
Yeah, I think you're referring to the work of Gao, if I'm not mistaken, like where he performed this, like in mice, he observed this ramping activity, and then with dentate nucleus cooling, this ramp was absent, and then there was like these planning errors in subsequent leaking movements in mice or rats, if I'm not mistaken. Yes, so I agree that that's probably related to that, and also like the fact that the motor preparation as a broader kind of perspective of discussion, what it essentially means. The preparation of the movement includes the postural components as well, like which portion of the limb to drive, especially the proximal parts. So I think that's also probably coming into play, that you have to have this postural preparation to perform the upcoming movement. So if the postural preparation is incomplete or unable to be performed, then the upcoming movement itself is hampered. So it's a cumulative error that might be happening over there. But yeah, so that's something that now we are like overall like shifting to looking at the motor planning part. So even before the movement, what is it that is going wrong that gives rise to these subsequent cumulative errors? So yeah.
I can ask one more question. What do you think might be the differences between targets 5 to 8 and 1 to 4? Because as you pointed out, from an inverse dynamics perspective, we would have expected something similar. But what's different about reaching out and toward the body that you think might account for the results?
Yeah, so that's the thing that, like, right now, again, I have a bit of hand waving. Laureen is raising a hand, I think. Maybe do you have some?
Sorry, I wanted to ask a question after, and it takes me a while to find the hand. That's all. Sorry about that.
Okay. All right. Yeah, so right now, to briefly answer your question, right now I don't have a great explanation other than doing hand waving, like because my the results are like I don't have anything to support or counteract like based on my data. So, but I think what is like as I said, like the caveats is probably the cerebellar output, at least in primates, like non-human primates. There might be a greater component of drive to the spinal cord itself, bypassing the transcortical normal route that is important in humans or evolutionarily becoming more important in humans. For example, the cerebello rubrospinal tract, which has and the innovation in the spinal interneurons could be biased towards like the flexors or predominantly the shoulder flexors, which kind of could give rise to these like differential effects on the reaching versus the retrieval movements. Other than that, I don't have, because going through the literature, I couldn't really find a lot of, because people, mostly the people that have performed these kind of studies in associated with weakness or asthenia or hypotonia, I don't think they performed any systematic like spatial analysis of the movements. It was mostly like clinical descriptions from like very old literature, also in the non-human primates. So yeah, I mean, I'm still looking for like anecdotal evidence or like, exploring this point now. Yeah.
All right. Laureline, you have your hand up.
Yes.
Hi, thank you. Thank you for the great talk. I have two questions. The first one is just a small clarification that maybe I missed. I saw that you had a plot about movement variability under cerebellar block for the planar movements. But at least in your schematic, it looks like the ending was very precise, even with cerebellar block. Do you see?
No, so it's, I think the way we have, I'm sorry for not being clear on that. So essentially what we do, so we are not measuring endpoint error here. So endpoint error is another phenomenon. In this case, I haven't looked there. So what we did over here before analyzing the trajectories, we basically aligned them from the start to the endpoint so that they allow, so because I wanted to look at like how from trial to trial, the trajectories are, how different they are, how unique they are. So yes, the one way of looking at it could be also endpoint error, because the endpoint error will also probably become more variable, like as there is overreaching or underreaching. But again, like to add to that, like the way we define the end of the movement is when the monkey enters the peripheral target. So I think the estimation of the endpoint is not as good as the trajectory errors that I have analyzed. So that's why I looked at it. But yes, to answer your question, that's just how we analyze the data. So there is more variability in the endpoint. Yeah. And
Thank you very much. If possible, yes. So it looks like there are really two types of hypotheses that you're looking at. One of it is pure muscle weakness, perhaps, that you've lost part of the drive. And then another one is, you discussed a lot state estimation. And so I'm wondering if you think that there would be ways of measuring the difficulty of state estimation in different reach directions. So is there any correlation between the effect of the cerebellar block and, for instance, how difficult it is to have a good estimation of your limb position based on proprioception for different targets? Thank you.
Yeah, so this, I think this work has been part of, like some work on this regard has been done by Amy Bastian, if I'm not mistaken. So they did this similar work in keen arm with control, like individuals like without cerebellar deficits, and compared them to cerebellar deficits, trying to estimate the location of the endpoint of the hand with and without visual feedback. So they also observe like this kind of proprioceptive deficits there. So I think this is so this is to answer your question. Yes, this is possible by like so they did it in two ways actually. Like one is like passively putting the limb in one location and asking the subject to locate the limb, and then also performing an active movement without visual feedback, and then also locating the limb. And then they found some interesting results there. So yeah, so this is something also that potentially we could explore in our model of cerebellar block. Thank you.
Thank you.
Nirvik, thank you so much. Thank you all. See you later.
Thank you so much. Appreciate your time. Bye.